Hello, everyone. Thanks for joining us today for the Advanced Drug Discovery and Development 2024 Online Symposium. My name is George. I'm excited to be here today alongside my colleagues, Professor Senna and Hannah James. Before we start the course today, I'd like to share and a few tips for the event. We would like to first of all ask if you could please refrain from video, audio, or photo recording of all the sessions across this event. Many of our speakers have kindly agreed to share their slides with you, and these are available within the literature section on the platform. Please also remember that we encourage debate and discussion and welcome all of you to use our networking services to introduce yourselves to other attendees, but we do not permit the overt solicitation of products or services and reserve the right to block any user who infringes on this rule. It's also incredibly important to say that this event was only made possible thanks to the generosity of our sponsors and partners, particularly our platinum sponsor, Lonza. For more detailed instructions on how to make the most of the features available to you at this event, please navigate to the event home and watch the overview video at the top of the page or visit the FAQ section in the left-hand menu. And with that out of the way, I will now move on to presenting our opening panel discussion for the event. We are about to dive into a critical and exciting discussion that touches on the cutting edge of drug discovery and development, one that is reshaping the entire pharmaceutical landscape. As we all know, the drug development process is long and complex, often plagued by challenges such as data limitations, accuracy concerns, and slow trial adaptations. But today we stand at the cusp of a revolution, one which is driven by next-generation technologies like artificial intelligence and machine learning. These transformative technologies are helping us overcome some of the most significant hurdles in drug discovery, from enhancing model accuracy to tackling small data challenges. Machine learning is also accelerating adaptive clinical trials, which is unlocking new possibilities for faster and more efficient clinical development. Within this panel, we are incredibly fortunate to have industry leaders who will share their unique insights into how AI and machine learning are reshaping the future of medicine. We'll explore not only the technical advancements, but also the growing interest and investment from major pharmaceutical companies in this space. So whether you're in research, clinical development, or simply passionate about the future of healthcare, I encourage you to stay engaged, ask questions, and be a part of this important conversation as we explore the potential of these groundbreaking innovations. So let's first begin by introducing our esteemed panelists. First of all, we have Adityo Prakash, who is the chairman and CEO of Verseon. Adityo started Verseon to change the way the world finds new medicines. He enjoys building fundamental science-based solutions to major business problems that impact society. Adityo has led the development of Verseon's drug discovery platform, novel drug pipeline, and overall business strategy. Previously, he was the CEO of Paulson Corporation and is the primary inventor of technology at the heart of all video streaming today. With a track record of delivering industry first, Adityo is an inventor on 40 patent families, and he received his BS in physics and mathematics from Caltech. Next within this panel, we have Dr. Raviv Pryluk, who is the CEO and co-founder of PhaseV. He is an engineer by training with a PhD in computational neuroscience and over a decade of experience as a technological leader in the advanced technology defense industry. He then transitioned into the health tech industry and joined Imuni, a biotech company harnessing single-cell multiomics and machine learning for drug discovery, as SVP of Operations and Analytics. Raviv then founded PhaseV to innovate the way that clinical trials are designed, analyzed, and executed, and is extremely passionate about the adaptive and innovative future of the drug development process, which will enable them to bring more treatments to more patients in a more efficient manner. It's an absolute pleasure to have you both join us today for this panel to open this event, and we're incredibly excited to hear your perspectives on the field of drug discovery and how AI and machine learning are revolutionizing the landscape for the better. So, with that out of the way, Adityo, thank you so much for being with us today. The floor is now yours to take us through the work being done at Verseon and how small data scenarios can be addressed in drug discovery and how AI is helping to overcome these challenges. And I believe that we have some slides prepared to help you showcase this to the audience.
Thank you, George. Hello, everyone. Let me tell you the story of how AI and other technologies are coming together to change small molecule drug discovery. I'll start my presentation by taking you through a quick summary of how small molecule drugs work in our bodies and how they're found. For those of you who don't think about these things every day, It's a good refresher. For others, it's something you already are probably intimately familiar with. Small molecule drugs are the bread and butter of the pharmaceutical industry. These low molecular weight compounds, organic compounds that can enter our cells easily and can interrupt disease processes. They're typically also administrable orally. And the way drugs work, when we take a pill, what happens to our bodies? What you see there are pictures of two proteins. Our DNA is just a recipe book for making these different proteins that fit together to form all our cell structures. But sometimes some of those proteins are associated with diseases. That protein drawn in blue on one side could be involved in a disease process, going and attaching to other proteins, cutting them up, etc. And you want to interrupt that process. To do that, you want to find basically one of these small organic molecules that can go bind in the pocket of this protein and prevent it from doing that damage. This is how every drug works, from traditional remedies that have been handed down to us to today's modern pharmaceutical miracles. But they're not very easy to find. How does the industry find drugs today? By pure brute force trial and error. It's a process called high throughput screening, where you take the disease-causing protein, you put it in these pipettes, and you stamp it down into each of these wells containing a different chemical. And you're looking for a change in color or temperature, and you say, aha, found a binder. Often they're not the final drugs. You have to spend the next 4 or 5 years tweaking the molecule to ensure that not only does it bind to the disease-causing protein in question, but will have better properties in the body. So, how many things can you test this way in a pharma lab? Anything you want to test must first be made. And humanity has been able to make about 7 million distinct families of compounds in the last 150 years, well less than 10, right? So across all of the pharma labs across the world, we're testing a max of less than 10 million distinct types of chemicals, right? But is that a good enough number? I think this next graphic puts it in context. That is a minuscule, insignificant fraction of the total number of possibilities that the rules of organic chemistry allow that you can make as possible drug-like molecules. There's a whole chemical universe that remains completely unexplored today. The troubling part is, when you think about it, is the fact that most of the great new medicines of the future lie in that unexplored chemical universe. And it has taken us 150 years to get to the 10 million that we or less that we have made so far. And the way AI works is that it needs a lot of data for training. Then when you ask for something similar, it knows what to predict. So AI, you can only train it on the available experimental data on things that we have already made and tested. And based on that, when you train AI, it can help you tweak known drug molecules. This is what's happening with all the current AI and medicine companies that have come along that are trying to use AI for making a difference in medicine. They're ending up producing these small little tweaks on old known drug molecules, because that's how AI works. It's not surprising. But this is no different from what medicinal chemists have been doing for the last 50 years. Vioxx versus Bextra, Lipitor versus Lescol, small little tweaks on somebody else's drug. And one could argue that AI might be able to speed up that process some. And as we say, we wish them luck, but the great medicines of the future won't be found this way. You need fundamentally new approaches, ideas, new structures that'll have remarkably different, properties in the body to treat diseases much better than we can today. But how do you search this chemical universe? If you have to do it by the true brute force trial and error process, it'll take us literally longer than the lifetime of the universe to, get there, right? To make all these different compounds and, try them. So the way to get there is differently, through computational means that do not require prior data like AI, okay? You can start with a protein structure in 3D on the computer and run against it billions of completely novel drug structures that you design on the computer that have never been made before, that are not available for testing. But to do that, you can't rely on AI because AI has never seen any of these molecules and does not know whether it'll bind and how it'll behave. You need to be able to use molecular physics. And you need actually some fundamental groundbreaking advances in molecular physics to get to the accuracy necessary where you can just replace the experiments that I just showed you and be able to, on the computer, design these things and figure out how it'll bind to the protein in question. If you think about it, this is how we design just about everything else in our bodies, right? Everything else in our world. Whether you build buildings, airplanes, computer chips, we simulate and design them on the computer first. Well, this is where drug discovery needs to go, where you can atom by atom design these completely novel drugs that'll do the perfect job that have never been made or seen before. Once you do that, then you can actually make them in the lab. This is what it allows you to find these clusters of potential drug candidates across the chemical universe. Once you find a bunch that are useful for a particular drug program, you make them in the lab and use that data to train your AI. This is what we do. Okay. Now, this AI that we have built is being trained on data that's not available anywhere else. The physics-driven process is generating new data, in AI parlance, what you call synthetic data. And then we are converting them to actual lab data and feeding all of that to our AI tools. But even within one of these clusters that you have found through physics processes and then made in the lab, it's not possible to generate enough dense data sets to qualify as what we call big data that's needed for AI technologies like deep learning. All right. For all of those, because of that situation, we needed to build something, novel AI technologies that work better in these small data scenarios. Here's a quote from Andrew Ng, who founded Google Brain. He says the number of teams that can build good AI systems from big data is in the hundreds, but a much smaller number can build good AI with small data. Well, he's right. And we have built technology that works far, far better than typical deep learning tools for these small sparse data settings, because real life data is very rarely big data. The term big data, as it says, is thrown around everywhere nowadays, and deep learning is the culmination of that big data approach to AI. It has great use cases. ChatGPT or other large language models, you can train them on billions of web pages. Face recognition models on hundreds of millions of photos. But these big data scenarios are the exception, not the rule. And today, most AI companies struggle to get their hands on enough high-quality training data. For all of these situations, we built what we call Verse AI. Small molecule drug discovery happens to be one of those kinds of examples. And what sets it apart is its accuracy prediction. Here, lower is better. You see 35% lower error rates than state-of-the-art deep learning technologies like Google or ML. This is what we needed to be able to have our AI tools perform far better in the small, sparse data settings so common for drug discovery. But it's also something that's can be applicable in many, many other settings in real life where you have small, sparse data. Most AI companies today, they don't really build their AI technology. They pull down software libraries from Google, Meta, other places, okay? But we belong in a different category. And this fundamental new AI technology that we've built dramatically improves the utility and reliability of AI in many of these everyday life settings where you don't have dense data, okay? When we combine our physics breakthroughs with these novel AI tools exploring, completely new drug structures, we see that we can find drugs with remarkable new properties. Okay, So I'll give you a few examples. Heart attacks and strokes, number one cause of death and disability on the planet, right? We have drugs that show far lower bleeding risk than any of the current drugs in use, which are some of the highest prescribed drugs on the planet, okay, to prevent these undesirable clots from forming that lead to heart attacks and strokes. Our drugs do not increase any bleeding risk, but prevent those clots from forming. Something like this has not been possible in the industry till now, and will actually positively impact the lives of almost 500 million people around the world. Okay, first drug passed phase 1, second one ready to enter clinical trials, and I'll show you another example. If somebody has diabetes long enough, chances are they're going to lose their eyesight. Okay. What you see on the right are pictures of a normal retina. The middle one looks bright because all the small blood vessels in the diabetic leak fluids, and that's what makes them go blind. We can take animals with raging diabetes, treat them with our drug, and completely stop the leakage and reverse the disease. We're the only company that has ever been able to show these results, and the best part is an oral pill. It'll change the lives of 180 million patients around the planet, okay? And that's just for starters. In the same fluid leakage is why diabetics also lose their other end organs, from kidneys failing and then having to go on dialysis to a range of other problems. And we can stop all of that as we expand the label to go after all other diabetic end organ damage. In that case, now the target addressable market is 550 million patients. What we're showing you are remarkable new drugs with uniquely desirable therapeutic properties that you can't find through current methods. You have to be able to explore this uncharted chemical ocean. Here is another example. The worst news cancer patients' families hear is that the drug has stopped working because you're giving them a drug, and the tumor mutates and the drug no longer works. We have developed, novel chemo agents that do not fall prey to drug resistance. The tumors can mutate, our drugs keep working. Program after program, what we're showing you are drugs, as I said, that can only be found if we can explore this uncharted chemical space. We have 16 different drug candidates so far, each with uniquely desirable therapeutic profiles that promise to change the standard of care for the diseases they address, okay? Ultimately, for the future of medicine, humanity needs to be able to explore this uncharted chemical ocean. Okay, this is where the great new drugs of the future lie. I gave you some examples of what's possible. Okay, and this is why also really being able to use AI in conjunction with advances in many other fields, because AI by itself is not a panacea. One needs to understand it's a useful tool. You combine it with other tools, and you also improve AI in ways that that's required, can really change what the world can expect from medicine.
Thank you, Raviv. Oh, sorry. Thank you, Adityo, so much for those fascinating insights into AI and small molecule drug discovery. And I'm sure that our audience is eager to learn more about the potential of AI in solving small data issues and improving drug discovery models. Raviv, based on your expertise with machine learning in clinical trials, do you see any parallels between the challenges of small data in drug discovery and those that are faced in clinical development? And how might AI-driven solutions in drug discovery apply to clinical trials?
Yeah, so I see both similarities, but also differences. And you would see in a moment throughout my presentation what is similar and what is different. I think validation piece, as Adityo was saying, being able to validate, to see the accuracy, the predictions of the models, and making sure that we are not only training, but actually validating and making sure that the insights are generalizable. This is definitely a core component that is shared between AI in discovery and development, as long as you want things to be meaningful and reliable. There are some differences in the sample size. You would see in a moment drug development is a unique area of small sample size versus big data approach, and it implies that we are talking about slightly different algorithms and slightly different models. I do think that the causal aspect, being able to find causality and not only correlation, is an additional thing that is shared between discovery and development. And last but not least, I do think that the future lays at the transition between discovery and development. So being able to translate what you've seen in discovery into what's applicable to human biology, this is where we are going to see the change. So
that's fantastic. Thank you so much, Raviv. Adityo, would you like to add any further thoughts or comments to that?
Completely agree with Raviv. Even when you find great new drugs, they have to be taken through clinical trials, right? And they're making the whole trial process far more efficient, being able to leverage patient data and use AI to be able to segment patient populations better, or do trial design better. That can have a big impact on the later stages of drug development. In the pharma parlance, we call what I just described as drug discovery. And what Raviv's company focuses on is drug development, meaning trials, making that far more efficient using AI. And there they have the benefit of being able to collect data over time from hopefully initially tens of thousands, then hundreds of thousands, and millions of patients, which will obviously have a huge impact on how efficiently you can design trials.
That's fantastic. Thank you so much, Adityo. Before we conclude this part of the session, Adityo, I just wanted to address an audience question that we've received surrounding your discussion, which could really contribute to the discussion itself. So we've seen AI successfully predict protein structures, but why did AI succeed in that context, but not in small molecule drug discovery?
A very good question. So here is the trouble with great big marketing announcements. AI is helping with protein structure determination, but protein, like I described the process of drug discovery, never did I say that finding the protein structure was the big problem, right? We already had protein structures for almost 97 or 98% of any disease humanity has been trying to go after because of lab experimental data, what's called crystal structures already done. So protein structures were never the big bottleneck in drug discovery. And AI has been able to help with protein structure determination for two reasons. One is that we had tons and tons of this experimental data across not just human proteins, but proteins from other species, et cetera, freely available, supported by big national efforts, in things like protein data bank, et cetera, so that you could train your AI models on that. There is one other thing. Protein structure often follows certain underlying biological rules. Okay, it's like a game where you are surmising the rules. You teach AI to learn how to play Go, chess, etc. by looking at protein structures across different species where function is preserved, and as a result, certain structures are preserved, even though the protein sequence is not exactly identical. The AI surmised those rules of the game, and it allows you to now fill out the remaining 2% of protein structures potentially that weren't in the protein data bank available yet. But we're often then going ahead and doing further experiments to validate if the protein structure is valid or not, that the AI is predicting. So that's fine, that's great, but that is an entirely different problem from finding a small molecule binder that will bind to the protein, a completely new structure that you have never seen. This has nothing to do with protein structure, and finding protein structure doesn't help you come up with these binders.
That's fantastic. Thank you so much for your expertise there, Adityo. Raviv, I just wanted to take a quick moment just to see if you had any thoughts or further insights to that question.
No, I think I completely agree with Adityo.
Excellent. Wonderful. In that case, thank you so much, Adityo, for sharing that detailed discussion on the use of AI in overcoming small data challenges in drug discovery. And now we'll shift gears slightly for the focus of this discussion and I'd like to welcome Raviv to the floor, who will talk about how machine learning is revolutionizing clinical development, including adaptive clinical trials and the future of this technology in this industry. And so, without further ado, over to you, Raviv.
Thank you, George. And thanks again, Adityo, for a great talk. So before I jump into the challenges and the solutions, let's talk briefly about the space. So Adityo was describing the preclinical work, how we are discovering new targets, how we are tailoring and finding how assets can target those biological assets. And this process is starting with many, many potential candidates, as we see here, but eventually we end up with handful of candidates. But only then we are starting the long and most expensive part, which is drug development. And there we have basically 4 phases. We have phase 1, where we are testing for safety. We have phase 2, where we are testing whether the drug is efficacious and can bring value for patients. In phase 3, this is the pivotal trials, where we are trying to prove and convince that indeed in a randomized control settings, RCTs, indeed the drug is working and doing what we've seen in phase 2. And this might sound odd to you, why we need to do phase 3 after we already demonstrated safety and efficacy. And surprisingly enough, so many drugs are failing in phase 3 despite being successful in phase 1 and 2. And we'll talk about that in a moment, what are the reasons for failures. But even after you approve the drug in a pivotal trial and you register the drug and the drug is in market, you have phase 4, when you need to show that the results are consistent between clinical trials and what we see in real-world data. And this is another interesting topic about why there are differences between clinical trial settings and real-world data. So this is basically drug development. This is a process that can take more than 10 years and can cost more than $3 billion, so quite long and expensive. And the failure rate there are enormously high. So you have a candidate that starts drug development process, there's less than 7% that this drug is going to reach out to market, which is super low. But why? Why so many drugs are failing in drug development? So I want to share with you 5 stories of failures that are representing quite well why clinical trials are failing so often. And then this is going to be the segue to tell you how AI, machine learning, software can be harnessed in order to overcome these failures and to bring new solutions in order to increase dramatically the throughput of clinical development. So the first story is about drugs that are almost significant. So drugs are working, but one of the things that you need to show is statistical significance. And if you can't show statistical significance, your drug is not going to be approved. So reasons for not showing statistical significance can be that you didn't design the trial appropriately, the sample size and the power of the trial were designed right from statistical standpoint. And this is an example from GSK, but it happens all the time. And there are those weird stories of like p-value of 0.053, and it's very devastating to have a drug that potentially could have been approved, but is actually not statistically significant. The second reason is that the drug is not targeting the right patient population. So the biology actually works, but we decided to focus on type 2 diabetes instead of type 1. We decided to focus on bladder cancer instead of non-small cell lung cancer. We decided to focus on older versus younger patients. We decided to focus on females versus males. That might be a better description of not the right population. So this is an example of a phase 2 trial, chronic cough, that we didn't target the right, the company, the sponsor didn't target the right patient population. And afterwards, this is going to be in the solution. When the right target population was targeted, actually the drug worked. And this is happening all the time. And now I mixed actually patient population and right indication. So this is another example. So if patient population can be specific biomarker or age or gender, right indication, as I was saying before, can be type 1, type 2 diabetes, or types of cancers, or in immunology today or neurodegenerative, we have so many indications, and not choosing the right indication can lead to failure. One very famous example, Viagra from Pfizer wasn't originally tested in Viagra, and then only after failing in another indication, Pfizer identified that actually the right indication should be Amigna. So this is another reason for failure. The fourth reason is not the right dose. So there's a delicate balance in choosing the right dose or dose schedule. You don't want it to be high enough because there are adverse events, and you don't want it to be low enough because you are not going to reach a efficacious response or binding between the molecule and the target. And there are so many examples of not choosing the right dose. By the way, recently in oncology, the FDA just released Project Optimus, pushing sponsors from finding the maximal tolerated dose that was under the assumption that actually the dose schedule or the dose curve is linear between efficacy and safety. Today we realize that we can increase dose and create more adverse events without bringing more efficacious response. So this is becoming more complex nonlinear curve that we need to identify, and for that we need to find different solutions. And last but not least, clinical trials can fail because the drug does not work, right? So there can be cases that it's not significant, not the right patient population, not the right indication, not the right dose. But it could be the case that the drug, despite efforts like Adityo and others did in the discovery phase, the drug is basically not working. So how are we going about overcoming these failures, right? So let's go one by one and tell you how AI and machine learning today, in general, and in phase V in particular, helping to overcome these failures. So almost significant, we developed a software tool for machine learning optimization for the trial design. In a click of a button using LLMs, we can harness the entire literature and scientific knowledge in order to see what happened in the past, to learn from past failures, and then using very sophisticated optimization algorithms to optimize the design. There are many parameters that we need to optimize. What's going to be the sample size? What's going to be the desired power? How many interim analyses are we conducting? What's the right dose schedule, and so on and so forth. So we are searching over this very complex space and finding the optimal parameters in order to increase dramatically the likelihood that if the drug works, we are going to see statistical significance results. In order to overcome the patient population challenge, we harness causal machine learning, and we'll show you in a moment some exciting results of when combining causal inference and machine learning, you can detect HTE, heterogeneous treatment effect, which is quite different than the average treatment effect. So different patients are responding differently if we're thinking about precision medicine era. So with these technologies, we can really detect the individual treatment effect in order to inform biomarker selection or subgroup detection. In order to deal with choosing the right indication, we developed sophisticated machine learning, biological and clinical modeling. So we are creating graphs that represent the biology in order to inform the indication selection and expansion process, and we would elaborate about this in a moment. In order to overcome the dose finding, we harness adaptive trials empowered by machine learning in order to find the right dose early on, already in phase 1, 2 trials, in order to inform afterwards phase 2, phase 3, and making sure that we are not going to fail down the road because we didn't find the right dose. And whenever the drug doesn't work, okay, it's not a magic, right? But if we can overcome the 4 first reasons, it's going to bring tremendous value for society and for the industry. So let's make things a bit more concrete. How is it done in practice? So when we are talking about ML optimization for trial design, this is a figure from our platform that can really map the space. And what you can see here is that we can 25x faster map the space and find the optimal parameters without compromising quality. One of the challenges whenever you want to design a trial, you run a lot of simulations in order to mimic how the trial is going to look like. With AI and machine learning, mapping the literature and then running millions and millions of scenarios and choosing the optimal design, becoming feasible and something that can be done in a click of a button in minutes, a process that could take weeks or months before can be now done in every biotech or pharma company with the Phase V platform. The causal machine learning, this is fundamental. We published recently quite a few papers on that. So basically, what you see here, this is a Phase 3 trial from type 2 diabetes. The trial originally failed, but what you can see here that actually 35% of the patients are responding very well to the treatment, and this is guided mainly by BMI and age. In other cases, it was genomic parameters. In other cases, it could be metabolic, previous medication. So any covariates, and we are collecting thousands and thousands of covariates. So the ability to detect the covariates that are driving heterogeneity and to inform the biomarker selection or the subgroup detection, this is at the heart of what the causal machine learning pipeline that we've built do. And this is serving today big pharmaceutical companies and biotech across the globe in order to increase the likelihood of transitioning between Phase 2 and Phase 3, in order to rescue failed trials, in order to inform real-world data interaction with clinical data, and so on and so forth. So this is another important example where we can harness AI and machine learning. The challenge of choosing the right indication, here you can see biological model in PD-L1. This is one of immunotherapies in oncology, but we've built those causal graphs in several therapeutic areas by now, in neurodegenerative like Parkinson's, and in immunology and oncology, as you see here. Whenever we understand the biology well enough, we can actually translate the biology into graph, and then translate the graphs into mathematical equations, and then eventually into synthetic data. As Adityo was saying, synthetic data informed by real biology is extremely beneficial in order to enrich the small data that we have in clinical trial. And by doing so, we can enrich dramatically and increase dramatically the likelihood that if there's a biologically meaningful signal, we can detect it. Not only that, we can start asking questions. What are the insights that we saw in Parkinson are telling us about Alzheimer's? What does the insights that we've seen from bladder cancer can inform us about non-small cell lung cancer, and so on and so forth. And by that, we can make the process of indication selection and expansion more informed, more data-driven, more biologically driven. In order to overcome the challenge of dose selection, here we harness adaptive trials empowered by machine learning and software. So adaptive trials are out there for many, many years, but because of the fact that they are less intuitive, harder to execute, harder to interpret, less sponsors are using those type of designs. But with the platform that we develop, in a click of a button, you can understand for various stakeholders, so for CleanOps, CleanDev, Biostats, commercial, to understand the implications of adaptive designs, to test millions of different scenarios and to see what's going to do to the power, to the sample size, to the statistical parameters, to the commercial, what's going to be the cost, what's going to be the time to market. And then in those finding studies, for example, you can design Bayesian trial, Bayesian optimal trials that are helping us to find the optimal dose, balancing not only safety considerations like adverse events, but also efficacy considerations, and to make it more streamlined. And the transition between design and execution is becoming more streamlined. And by incorporating this platform, we can increase dramatically the adoption of those adaptive trials that are out there for many, many years, but still not being used at scale. And this is exactly what we are trying to bring for the industry, and to make those finding studies more accurate, more reliable, more ethical for patients, to avoid administrating ineffective dose or even harmful dose. So it all sounds so fascinating and promising. Why it's not being done at scale, and why is it so difficult? So first of all, it requires advanced and unique technological expertise. It's not only expertise in AI and machine learning, it's also really understanding the unique aspects of clinical development, the biological implications, the clinical implications, the regulatory implications. You need to speak the regulatory language in order to make sure that things are interpretable and translatable. It's a very complex space to map and to understand and to trust. There is a trust question. We need to make sure that stakeholders can trust the findings that the AI and machine learning are informing us. And it requires a precise tailoring of the technology. So we are using technologies that were matured in other tech domains, in marketing and gaming and dynamic public transportation and econometrics. But clinical development is unique, and we need to make sure that the technology is tailored carefully in order to address the unique characteristics of clinical development. And to make it even more concrete, we need to address 5 fundamental challenges. We need to make sure that the AI machine learning algorithms are generalizable. So it's not enough to find the insights that are accurate in phase 2 trial. We need to make sure that whenever we are going to go to the phase 3 trial, the insights are going to be generalizable. We need to make sure that the insights are generalizable from clinical trials to real-world data. We need to make sure that the results are validated. This is a question that we are getting over and over again from sponsors. How do we know that this was validated? We are spending tons of hours and we are restless until we can know that something is validated. Internally with sophisticated statistical methods and machine learning methods, externally with additional data sets, with key opinion leaders, and every aspect and method that we can have in order to validate the findings. We need to make sure that the insights are explainable. The era of black box AI, I think, is not relevant for clinical development. We need to make sure that all stakeholders, clinical stakeholders, operational stakeholders, commercial stakeholders, executives, really understand and can explain what the model does. So it's not enough for the model to be accurate, it needs to be explainable. We need to make sure that it's actionable. We need to make sure that it falls under the regulatory requirements, that it's clinically meaningful, that physicians can really treat. It's not, an algorithm that in practice can work. So this is another major aspect. And we are spending a lot of time to make sure that the algorithms are actionable. And in some cases, we are paying toll in accuracy to make sure that things are actionable. And at the end of the day, you don't want to have cool algorithms in order to publish papers. You want to make sure that it's affecting patients' life. And last but not least, you want to have statistical guarantees. You want to make sure that you're speaking the statistical language that is so fundamental in clinical development. You want to have FDR, false discovery rate. You want to have p-values. You want to have assurance. So, and this is quite hard to translate machine learning and AI into the statistical language, and we developed quite unique expertise in doing this translation. So to wrap it all up, we at Phase V, we are a multidisciplinary team, and all that I was describing can be achieved only by a multidisciplinary team: software engineers, machine learning engineers, biostats, data scientists, but also physicians and biologists that have decades of experience in pharma and academy. We are headquartered in Boston, but operating globally. And here are some key members, the team, and happy to stop here and to take any questions.
That's wonderful. Thank you so much, Raviv, for that incredibly detailed presentation. It's truly exciting to see how machine learning is not only speeding up clinical trials, but also shaping the future of clinical development as a whole. Adityo, from your perspective on the AI side of things, how do you see these machine learning applications in clinical trials influencing drug discovery processes? Are there synergies that could benefit from both sides of the field?
Indeed. As I said before, discovery in vacuum is useless unless you can translate that to actionable results for patients. And to get there, you need to take them to trials. And making trials more efficient is really, really beneficial across the entire biopharma industry, because clinical trials, in my opinion, today the way they're run are some of the most cumbersome, ridiculous processes that humanity could ever devise. They're extremely long, extremely expensive to run, and very, very badly designed quite often, like the examples Raviv just gave. That arena requires a real overhaul. We've been, ourselves at Verseon have been thinking about it, looking for solutions that partners are developing, etc., because it is key to translating great new drugs that one discovers, into something that helps patients.
That's excellent. Thank you so much, Adityo. Raviv, do you have any follow-up thoughts on how these sort of technologies overlap between yours and Adityo's fields, I suppose?
So I think recently we started getting more and more requests to actually collaborate the insights and the models from the discovery phase into the development phase. Just to give one example, in those finding studies, phase 1, phase 1, 2 trials, you have a lot of data that you collected from preclinical models, and this can inform and serve as a prior for the design phase in the development. And whenever you have an accurate prior in a Bayesian framework where you're talking about priors and posteriors, as accurate as your prior, it's increasing the likelihood that your posterior is going to be accurate. It's going to decrease the required sample size, increase power, and this is extremely valuable. So making this process streamlined, taking models from discovery and incorporate them into the development, this is something that we recently started doing, and it brings quite a lot of value. But this is just one example, and I think that we are going to see more and more examples of how it's not going to be a siloed space where we have discovery AI and development AI. It's going to be more continuum approach toward removing the barriers between discovery and development.
One of the other things Raviv mentioned is explainability. I think that's critical for stakeholder adoption, but it's also actually really, really useful and critical for updating your own models relatively quickly and efficiently. I'm sure that's one of the reasons they've been focusing on this. And typical, Raviv, from what I understand, typical deep learning models, if you're using algorithms like SHAP, it's extremely cumbersome to try to tease out the explainability portion, right? Is that what you guys are doing, or you're developing something else?
So we have both. Whenever we can use off-the-shelf explainability like Shapley values and others, that's very valuable. But we also develop unique tools because there are unique characteristics. So we have layers for heterogeneity explainability to make it explainable already at the beginning of the process. Other unique either plots, figures, or models in order to make things explainable, reliable. So this is the heart of what we do, because we understand that various stakeholders have to understand, not need to understand, have to understand what's under the hood of the algorithms. This is core learning that we had. If we really want to change the way clinical trials are being designed and conducted, we need to make it reliable, explainable, and so on and so forth.
That's fantastic. Thank you so much both for those valuable points. And Raviv, before we conclude this session quickly, we've had an audience question that looks to be directed for you. So what is the biggest challenge in applying machine learning technology to clinical development?
So I think among these 5 challenges that I described, the trust, although it's not always clearly articulated, this is a major challenge, because there are very good reasons why we shouldn't trust machine learning at the core of what we do in clinical trials. We need to earn this trust. We need to create a lot of tools in order to convince various stakeholders why they should trust. This should come with a lot of hard work and a lot of trend test validation, a lot of explainability, a lot of experience. And I think for us as a company, we are gaining more and more use cases, and we can show to sponsors where it is working, where it's not working. Also talking about failures, it's not a magic, as I was saying. I think the fact that we can show sponsors that, for example, in the causal machine learning part, that there are some trials that we detect meaningful heterogeneity, whereas in others, we can come back to the sponsor and tell them, listen, I mean, we can find heterogeneity. I think this is reassuring and build trust, and I think this is an extremely important aspect.
Excellent. Thank you so much for these valuable insights into the latest advancements into the field of drug discovery. We're now going to move into the Q&A portion of our session. And a quick reminder to our audience to submit any questions you might have for Adityo or Raviv using the chat box on the right. And we will answer as many questions as we have time available for. But any questions that we don't have time to address will be sent to the relevant speaker after the event to be addressed offline. I can see that we've already received some fantastic questions, but don't hesitate to submit more as we continue through the session. The first question looks to be for you, Adityo. How does the use of AI in small molecule drug discovery differ from AI used in developing, say, biologics?
Good question. It's similar to the question you asked, someone asked about protein structure. Biologics are these large molecule drugs. Imagine I showed you these pictures of two proteins coming together and a small molecule drug binding to one of them, intervening in the disease process. Imagine instead you're designing another protein to bind to one of the proteins to intervene. Those are biologics. Those are proteins. The same reason you have more utility of AI in protein structure prediction. You can use AI for these biologics designs, okay, where you start with something and you want to make a small change. The AI, because it can do protein structure prediction, it can tell you, oh, okay, this one should work better, etc. So you have more utility in some ways of AI in these biologics design. So for the same reason, you have greater utility in protein structure prediction. You have a lot of underlying biological rules that these things follow, and you can actually leverage them. Biologics are obviously a very important component of the overall treatment, portfolio humanity needs to develop. But if there is a disease that requires hitting a protein target inside a cell, biologics don't work because those large molecule proteins cannot get through the cell membrane into the cell. So this is why small molecule drugs will remain sort of the bread and butter business of big pharma, and it'll be a very big part of how we treat disease.
That's a fantastic answer. Thank you so much, Adityo. Now we have one for you, Raviv. I know within your discussion you mentioned a lot that there were many key players involved in the landscape of using machine learning, but who would you say is more open to using these solutions in drug development, big pharma or biotech, and why do you think that is?
So I think actually today both, but it really depends, right? There are different risk proportions or considerations for big pharma and biotech. If you think about small biotech, usually they have one or few assets, and then they want to win, and they are willing to take risk to make sure that they are winning. For big pharma, failures are more painful, so in some cases they are more risk averse. But I actually think that based on our interaction and experience today in big pharma, we see a lot of excitement, truly excitement, about incorporating machine learning and AI carefully, as I was saying, whenever the algorithms are explainable and validated and differentiated. And I think that's what we are bringing. They are willing to incorporate them carefully because the competition is fierce. Today, I think big pharma understand that one of the CEO of big pharma told me, if 20 years ago when I was releasing a product into the market, I knew that the competitor is 2 to 3 years behind me. Today, it's 2 to 3 months. And with such a fierce competition, accelerating time to market, accelerating clinical trials, making them more accurate, more reliable, more ethical, is not a small thing. It's something that is essential for biotech and pharma. And that's why we see incredible traction and dialogue across the industry, across disease indications. So at the beginning, we thought that we are going to see it mainly in oncology and immunology, but we see it in neurodegenerative and endocrinology and rare diseases, obesity. So we really see it across the board. And I think it's a good signal. Obviously, it requires to continue building trust, keeping the highest technological standards. But when doing so rightly, I think sponsors would follow, because this is the right way to go.
That's fantastic. Thank you so much, Raviv. And we have time for a few more questions before we close out this session. And we have one from our audience that I believe both of you might be able to share some insights on. So how do you meet regulatory expectations with the use of AI and machine learning, since it is a greenfield area for many regulatory authorities?
So maybe I'll start by saying that first and foremost, I think we need to have open dialogue. And based on my experience, regulators, both in the US and in Europe, and also other regulatory agencies, are really open to have a productive dialogue. We are constantly seeing open forums and closed forums that we are participating in, and they are eager to learn and to consider the harm-benefit question. It's always about the harm-benefit, right? Whenever the benefit is high enough and the potential harm is low, they are willing to incorporate innovation. So it's our responsibility collectively as an industry to make sure that the harm-benefit balance is being maintained, and the benefit is high and the harm is low. And I think for that, the 5 key challenges that needs to overcome that I mentioned in my talk are extremely important to make things validated and explainable, and to have statistical guarantees. All these aspects are contributing to the dialogue with regulators. And I think it's clear for everyone that the future is going to incorporate AI and machine learning. It's only a question exactly where and exactly how.
From our viewpoint, early drug discovery all the way up to preclinical development, and when you enter clinical trials, up to that point, we operate in an arena where regulators don't, figure it all, because it doesn't matter if the drug fell on your lap, or you developed it through, some alien technology. All that matters is that have you been able to show in validated models of human organoids to animals to whatever else that it works, right? And so thankfully the early part of the discovery process doesn't involve regulators, but indeed as soon as you start entering clinical trials, the whole equation changes. And as Raviv put it, it's critical that we bring them along, so they start understanding and appreciating more and more a lot of these, not only the use of AI tools, but as a result, how adaptive trial design will keep changing, because a lot of the old style trial designs aren't really working, and you should have far better, more efficient ways of taking things to trials.
That's fantastic. Thank you both so much for your insights into that question. I believe we have time for one final question today, which is directed for you, Adityo. Are Verseon considering any aging drug candidates, and are there any that are in preclinical stages at all?
Yes, there are. They're not on our website. Yes, we are indeed looking at lifestyle drugs, anti-aging drugs. There are drugs that will help you retain muscle better automatically. Wouldn't that be nice? So yes, we're pretty excited about some of these other things that we're developing in the lab.
Thank you so much for that. And now that brings us to the end of our session. On behalf of Technology Networks, I want to sincerely thank both of our panelists, Adityo and Raviv, for sharing your expertise and for engaging in such a thought-provoking discussion. Today, we've explored some of the most pressing challenges in drug discovery and clinical development, from overcoming small data issues with AI to how machine learning is accelerating clinical trials. We've also touched upon the synergies between AI and applications in both drug discovery and clinical development. I'd also just like to take a moment to thank our audience for participating and for submitting such fascinating and excellent questions. If we weren't able to get to your question, don't worry, that all questions will be answered offline as soon as possible. So that just leaves me to say thank you once again, Adityo and Raviv, for your valuable insights, and to everyone in the audience for being a valuable part of this discussion. And we will be back soon with the next talk. Thank you.
Thank you.
Thank you.